tightness. Now, what I didn't tell you before, so far I've always talked about proteins that go through the membrane with alpha helices. Here's a different class, so-called beta barrels, and the word beta already sort of suggests it. They are strands. That is the outside here, and these strands essentially, each finger here is a strand. They are connected by hydrogen bonds again, and they form, the lipid is running this way here, and they form about a 45 angle. Is there some problem? No. They form, if that were the lipid bilayer, so that's outside, that's inside of the cell here, then they form a 45 angle and go really with these fingers and form, when you look at it from the top, I don't have a top view here, unfortunately. Well, it's a little bit of a sort of idea of a top view. When you look from the top, it actually is a hole. That's where the word barrel comes from. Now, and they do form pores in the outside cell. Pore essentially means that when it's big enough, anything that comes passes. Okay? And this, for some bacteria, that's why some bacteria have a second layer of membrane, because the first one is completely passive, lets molecules of that are not bigger than something through. And the second one has a sort of more controlled filter. The way now to look at them, again, there's an outside region here, an inside region, there's a loop inside, there's a strand that goes up, there's a strand that goes down, so down into the cell, up outside of the cell, there's an outside loop, and that again is a very simple grammar, if you want. And since it is a very simple grammar, you can again model it by some sort of hidden Markov model. How would you see whether the model that you model here makes sense? What could you do? The simplest thing. So essentially, it looks to you as if, when looking at this three-dimensional structure, there is some repetition, there's some pattern, and there's some rule that there's an upside, downside, and there's an outside loop, inside loop. It seems that they all do something like that. How would you check whether it's true? Well, this one, and this image, what you see here, is you label one. You point the inside loop to the inside loop, you point to the strand, next strand, and you can just trace it with your finger and see for that one protein whether that sort of rule system works. But now, how would you expand from there? Well, in principle, you can do it for many proteins because there are not that many proteins known that do this, so you can look at them all, but you somehow need to do counting. How would you count? Well, if that is your model, right, then you simply have to count at every single of these states. There's a loop inside, there's an upside strand, there's a downside strand, there's a loop outside. Count what you observe, right, in the structures that you know. And this one here gives the simple amino acid statistics, meaning it's simply the height of the letter here at particular positions of that strand. The height of the letter is proportional to what you actually observe. And the coloring reflects somehow the biophysical features, but this is outside, there's the pore, there's inside. What you see, those are the four states here, what you see immediately without spending much time looking at it, from these colors, one glimpse of an eye and you see the difference. Right? There is a lot of signal here, it may be not that easy to see in detail, but what you see is the four letter or the four ways of looking at these statistics and what you actually observe immediately shows you that the assumption that these are four different states and these four different states relate to the choosing or to the choice of four different types of sets of amino acids seems clear, right? Now, then the next step is essentially you take these observations that you have here in the amino acid alphabets until you translate them to the priors in an HMM. Prior essentially means what is your zeroth model? You don't have any assumption. So at least you sort of have the probability of all the residues forming that state. Okay? That's the zero, that's the prior. Oops. And that's, I didn't say that. By simply labeling proteins, in this particular case, the major problem was in doing the labeling here. And when you do that, then as the next step, you train the hidden Markov, beginning with these priors, and essentially you have a prediction model. And this prediction model, you then can see how often you are right in getting the upstrand, downstrand, the inner loop and the outer loop. Diagonal means you're right. Those are the raw numbers here of residues. Off diagonal is all mistakes. You see immediately that the diagonal is the highest. You also see that there are sometimes confusions here into different regions, and these confusions somehow make sense. But overall, it seems that this is I'm running out of batteries. So it seems that overall we have a relatively good performance, but the question is, how well can we actually distinguish between proteins that have barrels and those that don't? Okay, maybe if I know that something is a barrel protein, and all I need to do is put it into my model here, maybe that's easy. But maybe the proteins that somehow resemble barrel proteins locally, or have barrel domains, and don't have their barrel domains. Now, if I put something that has a barrel domain and also a non-barrel domain into this model here, and I don't know about domains, then I force the entire thing to fit. And when I force the entire thing to fit, maybe the model says, well, that doesn't work, right? So I have to cut it. And when I cut it, then immediately the question is, when do I begin to count something as a barrel? It's sort of one upside down, so it's one thread through the 4 states. Is that enough? Okay? And unfortunately, in terms of the biological reality, what we know is we have cases where, in fact, it is one thread through the model. So you have these 2 fingers, and from another chain come another 2 fingers, and from another chain comes another 2 fingers. And that builds up the barrel. So we have cases like this where you actually bring together 16 proteins to form one barrel. And they are not easy to recognize. But overall, that means we have to find a way to go from the per residue counting to going to a probability model that essentially addresses the issue, is a protein a membrane barrel protein or not? So I need some bit score here that assesses and so essentially what you see here is in dark accuracy or precision, in light the coverage or recall, and you see that for higher bit score, so if you're more certain, then you're more right, but then you find very few proteins, but you still find about 20% of the proteins at a cutoff at which you sort of seem to get 100% precision or accuracy. Where would you put the threshold? If you were to search an entire organism, an entire bacterium, you want to discover beta barrel proteins, where would you put the threshold? Let's put it like this. Would you put it there? In one of these two? Or what, one of this? Is that a good threshold? Is the second one here a better threshold? Or the third one? What would you do? So typically the kind of question asked my supervisor is not a bad answer. Ask the experimental biologist what they want. It's not a bad answer. The problem is you have to limit those kind of questions to the minimal. Meaning in your career of doing your master thesis, maybe you can ask the experimental biologist twice. but not more often. And this type of question you have coming up in your thesis every day. So 6 months times 30 is 180 questions you're not allowed to ask. So for 78 of those, you have to find your own answer. How would you do that here? And when you do your master thesis, you will not have much more knowledge than you have now. What would you do? Any idea? Any suggestion? Yeah?
I would put the threshold where the 290%, because you have around 70% coverage and about 70% accuracy.
Now, I'm going to do something that I really don't like to do, but I have to do that. Thank you for leaning your head out and making that point. The reality is that this answer is one that you find most frequent in the literature. This is what most people would do. They see that these two lines, recall and precision, coverage and accuracy, intersect, and by two lines intersecting, it appears, I guess, now is my interpretation, that there is some special point. There's no other logic to this choice than the intersection of lines. And I believe I take that if you don't have anything better to say. Again, I repeat, I believe almost every single student in my lab has given an answer like that, and that's not the only thing. There are many publications out there where people have done exactly that. But is there a better way of doing it? So let me repeat it differently. If you made that decision by choosing a point where two lines intersect, you possibly will find favorable reviewers because this is the most likely answer to find, including by very well-known scientists. Yes?
Wouldn't it make sense to go slightly to the right, but the coverage stays the same and accuracy goes up?
So that's the intersection point, and that's an interesting one. So I believe this is a point that this is the kind of thing that I try to induce here. I feel this is a very strong argument. If you trust, so you always have to say, my assumption is that this curve is representative of the future, right? But this is an assumption that you always put in for whatever decision you make, right? Whether you put the intersection line or any other. If you trust the curve, then this point is such that moving right is affordable at no cost. You do not lose in coverage, but you do gain in accuracy. So you should immediately move to the right. That's sort of a minimal, if you began with the intersection point, move to the right is sort of the minimal thing you should do by thinking through it. Yes, I agree. Any other point? So now, I've told you, I believe before, how difficult it is to experimentally determine something about a protein. So you may in fact get the sequence of a protein, but if you want to do the structure, you need to have it at a concentration at which you do not typically get it. That's not easy to do. Meaning that maybe you have a method here that took you a master thesis to develop, and the experimental biologist who has to follow up on this will most likely take a PhD thesis and more to follow up on a few of the ones you pick out. So if you found from grabbing into the soil here, found a bacterium, and you wanted to look at the most promising new membrane proteins in that bacterium, maybe the experimental PhD is able to do 3 or 4, maybe 1. But certainly not 50 or 100, okay? So what does that mean for choosing the threshold? Yeah?
The accuracy should be more important than the coverage.
So you have to clearly move somewhere up. And the most extreme way up is where you believe you don't make a mistake. Now we get again into this sort of logic that you applied before, where we somehow begin to see, okay, here's 100%, here is sub 100%, and it's the jump in cover. So essentially, thank you. That means you you want to move to the right, and then the question becomes between this point and that point or some point like that. Maybe the jump in coverage is so high and the loss in accuracy, this is the same kind of argument that we had here. You gain a lot on one end and don't lose that much on the other one. But ultimately, you will be inclined to moving the threshold to the right. We actually chose this value. So the leftmost point where we believe we don't make a mistake, we never believe our estimates well enough, so we believe we would make mistakes, and try to push it as much as possible. Okay, let's move into a new feature. Solvent accessibility is also a 1D feature that we get from 3D structures. One way of defining solvent accessibility, so if you had these 7 residues here, A to G, and you roll a ball of water over it. So this is the inside of the molecule. The blue is where the water can access the surface of the protein, right? The water ball essentially can roll. Inside here I have a blue, although the water ball itself cannot get there. So there is solvent in there, there's a sort of hole in there, but it's wrong, there's a hole. So solvent cannot get in because the water molecule cannot get in there. So I had also removed the blue here. And similarly here. But in this particular image here, residue C, and only the residue C is completely not accessible to water. But you do see that all the other 6 residues have a different degree in which they can be accessible to water. And that, in fact, is the absolute accessible surface. It's in square angstrom. So angstrom is 10 minus 10 is 10 nanometers. So it's essentially what square angstrom you can access by. This is one way of defining it. Now, when you join amino acids into proteins, you form peptide bonds. You know that. And in doing this, you have the side chains, and some of these side chains are longer than others. Some amino acids have very short side chains. Here, the alanine, for instance, and some have extremely long side chains, such as the lysine here. Now, that means that some of these amino acids have a different absolute solvent accessibility. So the long side chain has a long reach to being surrounded by water. The alanine short one doesn't. So the max for these different things will be very different for long or short side chains. That means that essentially long side chains may appear to be more water accessible. Even when the long side chains are somehow, in this particular case here, the C would have an absolute higher, if the C were a lysine and the B were an alanine, then the C, in this particular example here that I sketch, would have a higher absolute solvent accessibility than the B. Although the B is closer to solvent. So if you wanted to use the predicted solvent accessibility or the observed solvent accessibility to sort of arrange which of the 6 residues is most inside, by using the absolute number, you would make a mistake. You would put the B under the C. So under, I mean more inside of the sphere. So if you imagine a protein to be a sphere, or slightly elongated sphere. So that means you somehow need to look at a different number or a different way of doing it, in which you consider the maximal surface that you can have. So you have something that is comparable between alanine and lysine, between things with a short side chain and things with a long side chain. Essentially a percentage. So you ask what fraction of your side chain is covered by water. What fraction? And if you do that, I'm not sure I showed the example, but if you do that, and we went back to this example here, in this particular case, with a percentage, you would correctly classify that the B is more outside than the C. Now, you can also begin to look at states. Looking at states, a binary system, you have something that's in the inside, buried, and something that is exposed to solvent. And then there are publications that talk about buried intermediate exposed. What is best? When we develop a prediction method, how would we do that? Again, your first answer maybe I Google and I see what others do. Perfectly valid answer. And most of you should do that. Most students during their master thesis don't do this enough. Do more of it than you are inclined to, is my suggestion. But now more scientifically, yes? Yeah?
Maybe you use some cross-validation or cross-ranking approach?
So that's a great idea. So essentially you ask, you don't answer this, but you simply try different prediction methods. You try all these alternatives, and you see what can you predict better. And there is some rationality. Now, the problem becomes here, yeah? No. You wanted to say something? Or?
Yeah,
After. The problem becomes, what do you call better? Okay, let's just look at the simplest example here. In the simplest example, where we have the alternative between using 2 states, buried exposed, and 3 states, buried intermediate exposed. So one sort of is trivially 50% accurate, and one is trivially 33% accurate. So if the states would be equally distributed, right? So we cannot say by the number being higher, this works better. So you have to somehow measure an improvement over random. So the absolute number will not be a criterion for improving it, but if you have a better improvement over random, then you would argue, well, say 3-state solution is better, or percentage is best. And that, I believe, is ultimately the way to go. Another way you could do that is you look at what is more conserved in evolution. Because you assume that what's more conserved in evolution is more relevant for the details about protein structure and function that you try to capture, but you're not capturing directly. Your point?
that kind of answered my question. That is, how does one define a state? Like, what is the threshold between buried and exposed? Like, how is it?
And before I go into let me answer that question directly. So there are many papers. So first of all, historically, the first thing was to define the threshold according to square angstrom, simply because that was the first measurement, the first way in which people measured solvent accessibility. Relative solvent accessibility required further experiments that people did later, so that later thresholds on binary states were done on relative numbers. And now relative numbers, there are papers that say the number should not be 20. So if I did a 2-state model buried exposed, then the number should be 20. So lower than 20% is buried, more is exposed. And there are papers then that say it's not 20, it's 25, it's 9, 16. There's one paper from somebody who got very famous, meanwhile, that is arguing 7 is the right number. It's not 6 and it's not 8, it's 7. The people publish papers on all kinds of issues. That is one of those. But I ultimately believe that the answer that you gave before, what is conserved in evolution or what can be predicted, that should also be the answer here. A good way to distinguish this is if you can predict it. A good way is if it is evolutionarily conserved. Another way of good way would be if you could use it for the next step. The next step could be to build a 3D structure. So what in that context? So it's not only what can I predict now for this, but what can I use the prediction for? Why do I develop that prediction method? And so that may also help in determining the number. Now, when we look at it, one thing is clear, that whether something is 100% or 80% exposed, the difference between these two numbers is 20%, so 20 percentage points, right? This is not that different. So both of those essentially are exposed. They are on the surface. They are exposed to water. While 0 or 20%, that's a big difference. 0 means it's completely inside of the protein. 20% could be such as the D here, that is relatively inside, but it somehow still has a little bit of an angle to water. Or it could be a hole in a protein. It could be, imagine a protein to be a sphere that somehow has a dent in here. And this dent could exactly be the 20%, but it is still somehow inside, or it is under the dent finger here. So it's the one underneath here, which is pretty much inside. It sort of has a little bit of an exposure. So 20 or 0 is a big difference. 80 or 100 is not. Both of them are on the outside. And somehow, this is what I want to reflect in the number that I do. And the simplest one that we looked at was simply a square root. So where we essentially have 10 states, or an infinite number of states, but we essentially switch from percentage solvent accessibility to some state number or some state index by a square root. In this particular case, is simply translated to 10. And in this particular case, we really predict solvent accessibility in 10 states. Historically, the first way to predict solvent accessibility is again by hydrophobicity. Hydrophobic means inside. Hydro is water. Phobic is you want to stay away here of water, so you want to be inside, stay away from water. When simply hydrophobic residues are inside. Hydrophilic residues, Phil is friend, want to be close to water, they are at the outside. That's the way historically has been done. Later, but again for you guys, a long time ago in history, it was done by machine learning. And that machine learning, the first machine learning device that did it in exactly the way that I said, with the square root, where we, in this particular case, a neural network, has 10 output units. The first one codes for 0 to 1% solvent accessibility. The next for 1 to 4, 2 to is the square of the number of the unit. Okay, and this simple coding makes sure that between 0 and 1 there's a unit difference, and 81 to 100 essentially there's a 20% percentage points that do not really matter, right? So in this way of coding, the other advantage is you do not decide on how you put the threshold. So you have 10 output units. You essentially predict a value for percentage. Why did I not put in one unit that predicts percentage? Because it didn't work, or it didn't reflect. So I could have put the square root function directly into that unit. Didn't work. The approach with 10 units worked much better at the time. Now, since I can predict with this device the very number, I can choose my threshold according to other concerns in the future. In particular, I can let the user, for whatever project they will use it in the future, choose the threshold, what is buried exposed. And again, this turned out to work better than pre-choosing a threshold, at least for the thresholds that I looked at. There's also, again, this issue of reliability index. I talked about it for secondary structure prediction. It's the same thing here. Now is the open circles here, and that is projected onto a two-state system, buried, exposed. When I predict for all residues, then in a two-state Q2 accuracy is 74, roundabout, was 74. But I can also look at the 50% most strongly predicted residues here. And for those, the number goes from 74 to about 84. Okay? So just looking at how strong the prediction is and the strength of the prediction, this again is a typical kind of question that we have asked in the past, and we will keep asking in the future. And I'm going to show you only on a network with 2 output units. Here we have 10. The story is slightly more complicated, but it's in principle the same. So if your neural network has 2 output units, and say this is the one for buried, and this is the one for exposed, and say the value of this unit here is 0.8, and the value of that unit is 0.2, then you say that the residue is buried. If the value on this one is 0.6 and the one of that 0.4, you also say buried. Same decision, but in terms of the prediction there's no difference, but the difference is in this reliability index. So when we move to the left here in that direction, we look at only the fraction here, for instance the 50% for which this difference is highest in my data set, right? Now again, this is a number that you see from the prediction. The map to what accuracy it relates to That you only see for the proteins for which you know the structure. But the network, on the level of the network, you can decide where you are on this axis. So you don't need to have a lookup. You can do that for any prediction in the future, right? You can say how, or you can define to the user how reliable your prediction is, and then you have to provide this graph here to show how reliability translates to expected accuracy. That concept is clear to you? Yeah?
Could you please quickly explain again what the axis on this chart?
Thank you. And this is absolutely my mistake. Yes. Imagine that you now translate the difference between these into a probability. And on this probability axis, you can have a probability of making the decision, whatever decision you do. The decision is B or the decision is E, either of those two. But you have a certain reliability or probability of making that decision. I call it reliability. And the reliability can be 1, say between 1 and 0. Okay? And that 1 or 0 is for this, or that could be swapped state. Now, what I'm going to show on this axis here is exactly that number. From 0 to 1. So on the right-hand side of this graph, I show what is my performance if I'm forced to give a prediction for every single residue. Give a prediction for every single residue, I will simply take the highest number. So whenever my threshold is, whenever this number is higher than 0.5, it's 1 unit, otherwise it's the other unit, or whatever the detailed decision is, right? That's the default decision for every single residue by whatever is the largest. That's here. I make a prediction then for 100% of all the residues. This axis here shows the percentage of residues for which I make a prediction. If I allow myself to choose the ones where the difference has a certain minimal value, then I no longer make a prediction for all the residues. I just look at the ones for which the difference is above some threshold. And the higher the threshold I want this to be above, the fewer the residues I'm going to predict for. Again, the threshold is 0, I predict for all residues. The threshold is something that is called RI4 here. I simply map the difference here onto integers from 0 to 9 in order to have, when I look at it on, when I display the prediction in an ASCII format, I have a sequence, and then I have a B or E, and then I have a reliability index. I needed one symbol per letter. That's why I map it from 0 to 9. And on this map from 0 to 9, 9 is the highest, 0 is the lowest. Essentially, you can think about it as a threshold of 0, a threshold of 9. And again, for the threshold of 9, you have, in this particular case here, threshold of 9, you have very few for which you still make a prediction, because for very few cases, the difference is that high. Okay? That's why in this direction you have fewer. Now, on this axis, I show the percentage, the precision or the percentage accuracies. In this particular case, really, it's a two-state accuracy, because I have the two states buried exposed. And in this particular case, underneath is 16% as a threshold, incidentally. And I simply ask, what's the number for the subset that I'm looking at? You see that the line here, that's solvent accessibility, that incidentally is for secondary structure. So this is actually a three-state model here. But you see that these lines go up. Things that are predicted more reliably have a higher accuracy. Well, I'm very happy about that. But it's not necessarily what you observe. We have developed machine learning devices where this was actually flat. Where we, in fact, have tried to put the signal of stronger prediction in, but we couldn't use it. Typically, that implies that something goes wrong. When your machine learning device learns such a sort of gray, or such a white and black classification, no gray line in between, typically something went wrong. Here, this is one of the sort of good ones, but you know, this is not untypical. Okay? Is the curve clear? Or is the data clear that I show? Thank you for asking, by the way. Yeah.
Basically, when I look at the leftmost empty circle, it would mean I make predictions only for residues that give me this RI index of 9.
Yeah, this is 9, 8, yeah.
Yeah, I could maybe predict about 10% of my residues, but
Yes.
with 90% accuracy.
Above 90% accuracy, exactly. And again, the beauty of this plot is that you can tell the user what those are. You can look, without knowing the answer, you can look at the prediction and simply say, label, those are the 10% strongest, most strongly predicted residues. We believe that for those you have an accuracy and expected precision above 90%. Okay? This plot here, you need structures for. You need to have labeled data. The label on this is the 10% strongest, you don't need a label. You simply can predict that. That is part of your prediction model. That's simply the output you observe. Here is the output you observe, right? So this axis you have in a real prediction case, bless you. This axis you only have for the cross-validation, for the data that you initially, then you give that plot to the user and, or in some way, and you tell them, you pick that point, this is your expectation. When you look at all residues, this is the expectation. When you look at the 10% strongest, this is the expectation. Okay? Clear? It is a cumulative plot, I have to say. So, meaning that the number here is not the residues, or the number here is not all the residues with a difference of 4, but this is with all the residues of a difference 4 and higher. Okay? That is what I mean by cumulative. Did I create more clarity or more confusion? Somewhere there are clusters of clarity, and the confusion clusters are too confused. Is my interpretation of what I see. Now, detailed predictions, meaning the percentage solvent accessibility, there is some issue, but clearly these methods do very well in a coarse-grained two-state model, buried versus exposed. What do I get from these predictions? One example is shown here in a method that is from the Nir Ben-Tal group, Tel Aviv University, Conserve. This, he takes structures that you know, and this is just one particular protein structure, and the label, the coloring here is according to conserved in red, and less conserved, more variable, blue. And this here, well, now if you were more used to looking at protein structures, you would essentially see that this looks like a binding site. This is the kind of hole that the substrate will bind into. Here the substrate, in fact, is DNA. And you can immediately see the binding site. You cannot see the binding site if I remove the colors. All you need to put on top here is the conservation in an alignment, in a profile, in a PSSM, in a family. And now this you can do for a protein of known structure, but for the kind of prediction methods that I showed, and Nir Ben-Tal's group has been using that too, you can also do it for proteins for which you do not know the structure. And simply predict the surface, put the conservation, and use that to find cavities. In this particular case, we have a cavity that is not conserved, or conserved cavities that have different substrate. And you can see this. We can use the predicted solvent accessibility. This is a slightly outdated slide. I said you can use it to predict subcell localization. You can use it to predict protein-protein interaction, flexibility of motion of structures. It has been used for all of those, and many more. So there's a large battery of methods out there that use solvent accessibility and secondary structure prediction as input to predict other aspects of protein function. And typically, so the way this lecture is structured, the part that comes in the winter semester is the part where we look at the type of methods used in order to predict more directly function that use these features or these methods that I described in the first lecture as input. Here's another way in which we can do that, and we're getting back to the question that you phrased correctly in some sense for today's lecture, which is to identify domains. It's funny that you began with that very question of domains, and we end this lecture with that. So the way, again, to think about structural domains is there are folding on their own three-dimensional structure, they're somehow compact, meaning this kind of thing is a domain, while this one here does just not have enough internal connections. So I would argue this is a fragment. This may be something that has a domain and a little bit put on top. It's called chain. So this is what we often observe in proteins, that there are additional loops put on top of it. There is a domain underneath. The protein exists as the blue one here, so where you have some additional, let's call it embellishment. So in addition to the globular soluble domain. And then there are sort of things like that. Now the question is, how can we, when I give you a sequence, see what we are related to? And the simplest model really would be to simply predict the residues on the surface. So you take a two-state model, you take whatever sequence you have, you predict the number of buried and exposed residues, and you see whether it fit, to which of these does it fit best. And all you need to do is essentially assume that proteins are spheres, or slightly elongated spheres, and you can simply work out a relation between the length of a protein and the number of exposed residues. So if you assume that the protein, all residues were the same size and the protein would be a cube, that's the line of cubes here. If you assume it's spheres, then that is what you get. And the blue is what you observe. So you will see that the observation is very close to assuming that the protein is a sphere. Okay? And this is really a totally simple model. You can program that walking from here to computer science. Actually, it took me a little bit longer to sort out the equation, but it was on a walk not much longer than this. So it really is simple. So getting the data, this is more complicated, the blue part. But if that is such a good fit, then you can simply predict and see where you don't fit. And use that to predict regions that are not looking like real globular protein domains. Okay.